All right, welcome. My name is Morten Knudsen. I'm an associate professor at the Center for Healthy Aging at the University of Copenhagen, and I'm going to tell you about genome maintenance and longevity. So my group focuses on understanding aging and understanding the phenotypical traits that occur with age. So, for example, try to figure out why we develop graying of hair or facial wrinkles, and basically why we get old, and try to understand the molecular basis of these phenotypes with aging, and then try to develop interventions to deal with these phenotypes. And so, many things happens when you get old. So here we have the prevalence of features that occur in an elderly population based on, basically we downloaded PubMed. So we can see the graying of hair is highly prevalent. About 99% of everybody will develop some sort of graying of hair. Muscle weakness is also highly prevalent in the elderly population. And then there are more sort of serious or pathological conditions that occur with aging. So for example, about 40% of everybody will develop some sort of cancer. And between 10 and 20% of everybody will develop some sort of dementia. And so what drives this phenotypical variability is not really well understood. So in today's lesson, we will try to understand how DNA damage impacts aging and the phenotypes associated with aging, and also what we can do about this. So let's first define aging. I think the most simple definition is that it's a time-dependent phenomenon that increases the risk of death. And looking at the data, it looks something like this. So as you get older, your risk of dying the next year increases. And this is data from the Social Security Administration in the US. And as you approach 122 years of age, your risk of death approaches 1, because the world record of aging is 122 years. Looking at survival, and so this risk translates into a survival curve, and this is in one cohort of people. This is actually the Danish population, the survival curve of the entire Danish population. You can see that as we get older, fewer and fewer people are alive. This is the surviving fraction. And this graph in itself is not that interesting, but we can actually, because we have quite good registries in Denmark, we can look at compounds that may impact the survival curve. So each line here represents a group of people that have taken one prescription drug. And so you can see that certain drugs that are associated with longevity and certain drugs that are associated with shorter lifespan. And so one point is that aging here, it's not magic, that it actually can be influenced by, for example, environmental factors such as drug intake. But of course, aging occurs in multiple organisms. In the lab, we use humans, mice, and fruit flies. And the real difference between us and, for example, a fruit fly is really our genomes. And so returning to sort of the risk over time, here we have an organism called a hydra, which has the same constant risk of dying as a 19-year-old human. So in essence, this organism, per our definition at least, does not really age. So aging is not magic. Aging is also influenced by our genes. And in fact, you can get mutations in genes that can give you premature aging. So you can inherit some mutation from your parents that will unfortunately give you a phenotype where you age much faster than normal people. Interestingly, most of these mutations are actually found in DNA repair genes. So these are enzymes that help repairing DNA over time. And in fact, we know that with age, you accumulate markers of DNA damage. So here we see, this is in mice, we see that a marker gamma H2AX, these are the green blobs here, increase with increasing age in multiple tissues in mice. This also occurs in humans. And if you induce cellular senescence, so this is a phenotype where cells stop growing, then you see that that there are DNA damage foci in these cells that seem to persist over a very long time. And we will return to that later in this lecture. We also know with age that mutations actually increase in single neurons. If you do single cell sequencing on humans with age, you see this increase in mutations with age, suggesting that these could be part of the reason for why we develop age-associated neuropathologies. And in fact, you can inherit these defects DNA repair, and then that can lead to premature aging. And there's a large number of these diseases, and some correspond more closely with normal human aging than others. And so to understand these diseases better, we created a database, and here we have a disease called dyskeratosis congenita, where you can see the prevalence of features, just like I showed you the prevalence of features in normal aging. Here we just have the prevalence of features of patients that suffer from dyskeratosis congenita, for example. And you can see that 89% of patients have skin pigmentation changes, nail dystrophy, and leukoplakia. And dyskeratosis congenita is an interesting disease. It's caused by mutations in the telomerase complex. So these are enzymes that are associated with lengthening the telomeres and allowing cells to divide more. So I think what's quite obvious here is that only certain features of aging are seen in dyskeratosis congenita. You don't see, for example, neuropathologies. And maybe this makes sense because because neurons don't really divide, for example. But there are many other diseases. Telangiectasia has a different type of phenotypical spectrum. Patients suffer from radiosensitivity, so they are hypersensitive to ionizing radiation. Then they have issues with myelination in the brain, for example, attacks and so forth. Cockayne syndrome is a disease that we'll return to in a while. This has a large spectrum of features associated with aging, particularly issues with the brain. So because we have this quantified description of diseases, including a quantified description of aging, we can see how different diseases associated with each other. So here we have each column is a disease and each row is a phenotype. And then we can actually do hierarchical clustering. So, and then of course the red dots here represent the prevalence of that particular feature. And the darker, the higher the prevalence. So we can see the certain features associated with each other. So short stature, microcephaly, and developmental delay seem to co-segregate. This makes sense. These are probably related to defects in growth. There are other features that associate with each other. So sun sensitivity and cancer appear to be quite closely correlated. Features such as the neuropathology features, cerebral atrophy, attacks, neuropathy, also associate with each other. So we can figure out how the different features associate with each other, but we can also figure out how the different diseases associate with each other, because there's no real good understanding why a certain type of DNA damage leads to a certain type of pathological outcome. And so here we can, we show the hierarchical clustering again. We can see that there are some diseases that associate with each other. So the green here, I've labeled them based on the putative DNA repair deficiency. So there's a DNA repair pathway called base scission repair, this one called nucleotide scission repair, this double-stranded break repair, mismatch repair, and I also included just aging. And what you can see here is that the red colors are everywhere, the green colors are everywhere. the purple colors everywhere. So the take-home message is here that even though we know some of the enzymatic issues associated with these different genes that are mutated, we have no understanding really of how they then translate into the clinical phenotypes we see in the diseases. Another way to look at it is looking at the overlap and features between diseases like this. So each dot here is a disease, and connecting lines between dots indicate that the diseases are associated with each other. And here we also see that there's not a great correlation between the different diseases. We can also increase the threshold. And here maybe something comes out. You can see the purple diseases. These are base edition repair diseases do seem to maybe form more of a cluster, but it's not a clear cut. Again, highlighting that we have very limited understanding of how the enzymatic defects found in these diseases translates into a phenotype. So that all looks quite good, but what is it actually good for? Well, we can also include diseases where we actually know how or have a good idea of how the pathological event leads to the pathological phenotype. And so here we've clustered a couple of 100 diseases. And here we get obviously a large amount of different features, much larger than just looking at the specific DNA repair disorders. We can then actually cluster DNA repair disorders such as ataxia-telangiectasia, Cockayne syndrome, XPA. So the blue ones here so the red diseases here are primary mitochondrial disorders. So these are diseases where you see mitochondrial defects, and you can see that there's quite a large overlap between some of the premature aging diseases and mitochondrial diseases. The green ones are control diseases, where we do not think there's any mitochondrial involvement. So I'm going to talk a little bit about one of them, Cockayne syndrome, which is a premature aging disease, also called an accelerated aging disorder. The patients develop cachexia and growth retardation. They have ataxia, sensory hearing loss, seizures, muscle weakness, and lactic acidosis in the brain. They develop cerebellar, also cerebral atrophy and calcifications, and the patients also suffer from hypertension. We looked quite closely at the features of a mouse model of this that is mutated in the CSB gene. And so here we did an MRI scan of the mice, and what we can see is that, so the yellow here is fat, and you can see that with age, you tend to gain weight. Unless you suffer from this premature aging disease, then you're actually maintaining this quite skinny phenotype even into old age as a mouse. And we also saw that there was loss of cells in the spiral ganglia in the inner ear, which could explain the sensorineural hearing loss that you see in these mice and that you see with normal human aging. So we wondered if we could exacerbate the phenotype by feeding the mice different diets. So we have mice on a standard diet on caloric restriction, and we thought that the caloric restriction would be beneficial for the mice. We also had mice on a high-fat diet, and because we did the study with Rafa De Cabo, we also included resveratrol. To our surprise, the high-fat diet actually appeared to be very good for these mice. It normalized their body weight, it normalized the metabolism of the mice, it rescued the hearing loss, and it normalized the transcriptome of the cerebellum. And we actually did not see any adverse effect. The mice did not develop diabetes, for example. One thing we looked at was this increase in lactate production, which you see with normal human aging, and which you particularly see in Cockayne syndrome. So the diets didn't really do very much for that. We saw that there was increased lactate production or increased lactate levels in the circulation of the mice, and you had a higher level of extracellular acidification rates in fibroblast cell lines from a patient that has been compared. So this is a patient cell line where we overexpress wild type CSB or an empty vector. So this is the CSB deficient cell line, and you have much higher extracellular acidification rate. So just to remind you how lactate is produced, so glucose is converted to pyruvate through glycolysis, and pyruvate can then be transported to the mitochondria and converted into acetyl-CoA, or be converted into lactate. And so this conversion very much depends on the NAD/NADH ratio. So we next wondered there could be changes in that, and indeed, the changes in the NAD/NADH ratio actually completely explains the differences we see in lactate production. So this actually now became quite interesting for us because NAD is also a factor that's important in DNA repair through this enzyme called poly-ADP ribose polymerase 1, PARP1. So PARP1 polymerizes NAD. It actually consumes NAD as its enzymatic function and creates a polymer. And this polymer is then used to recruit other DNA repair enzymes and to open up the chromatin. And it's been shown to be involved in features of aging. And indeed, looking at this polymer, so here we're using a Western blot to look at the poly-ADP ribose polymer. We can see in the cerebellum of the mice, you have more of the polymer, and this corresponds with a decrease in SERT1 levels. In nematodes, we can see that with age, you get more of the polymer, and particularly in the CSB deficient nematodes, you have a lot more of the polymer. And this is also the case in the human fibroblasts. You have more polymer in CSB deficient cells and ATM deficient cells and in XPA deficient cells. And these three cell lines are the ones that have neurodegenerative features. And so the idea is that PARP activation leads to loss of NAD, and then that could lead to mitochondrial dysfunction. And indeed, if we inhibit PARP1, we reduce mitochondrial superoxide production, so reactive oxygen species from the mitochondria. We also normalize the membrane potential of mitochondria in CSB deficient mice if we give them a precursor for NAD, and we normalize reactive oxygen species generation in mitochondria isolated from the cerebellum of the mice. All right, but we still have not answered the question, how could the high-fat diet actually work? So we know that the brain does not metabolize fatty acids, so it could be a secondary metabolite. And one potential metabolite could, or group of metabolites could be ketones. So ketones are basically is synthesized by a breakdown of fatty acids. So fatty acids come either through the food intake or through fatty acids that are metabolized from adipocytes. If you're fasting, you get fatty acids. Fatty acids are transported to the liver. The liver will break down the fatty acids into acetyl-CoA, and two acetyl-CoA will then combine to form ketones, beta-hydroxybutyrate. And this will be primarily beta-hydroxybutyrate. This will be transported to the blood, and the brain can take up beta-hydroxybutyrate and convert it back to acetyl-CoA that can then be used for example, energy production or myelination of the brain. So one idea was, since we have this increased flux from glucose to lactate, you're lacking acetyl-CoA, and ketones can then go in and replenish acetyl-CoA that can then be used in the mitochondria. And so we actually measured acetyl-CoA, and our high-fat diet did increase acetyl-CoA levels. Particularly in the brain, acetyl-CoA levels were decreased in the CSB mice, and giving them a high-fat diet increased that. And we can treat CSB-deficient worms with beta-hydroxybutyrate and increase the lifespan. So the idea is that during healthy aging, you have you get DNA damage, but you have proficient DNA repair, allowing us to shut down PARP1. If you have a problem with repairing DNA, you lose NAD, then that leads to loss of acetyl-CoA and neurodegeneration. And in fact, we can use a PARP inhibitor to increase the lifespan of CSP deficient nematodes. We can also use an NAD precursor, and then that normalizes the entire transcriptome of the cerebellum. Or we can use ketones to normalize features, cellular features in CSP deficient cell lines. But why would PARP activation lead to specifically a neurodegenerative phenotype? So considering, I think we need to think a little bit about the energetic needs of the brain. So the brain is quite special as an organ, because many special or many processes have to occur. For example, in neurons, you have to have neuropeptide synthesis. There's synaptic interactions, so you have synaptic pruning and reformation throughout the life course of an organism. So you have these sort of relatively large structural changes that occur over time. But of course, neurons depolarize and repolarize again and again and again, and this is a highly energetically costly process. And so PARP activation is also actually quite highly energetically costly. Not only do you get this changes in the NAD/NADH ratio, which facilitates less transport of energy to mitochondria, you also have to resynthesize NAD. And this occurs through a cycle such as this. And you can see the net reaction here is that you convert for each NAD, you convert 2 ATP into 2 AMPs. So this corresponds to 4 high energy phosphates. So this is quite energetically costly. And indeed, if we look at the ATP half-life in cells, so at time 0, we're inhibiting ATP generation through glycolysis by using 2-deoxyglucose and mitochondria by using oligomycin. And we can see the half-life is much faster in CSP deficient cells. So if we sort of go back to the idea is that you have, under normal conditions, a equilibrium between supply and demand. If you have mitochondrial neurodegeneration, and I will remind you that the neurodegeneration that we see in these disorders is very similar to what you see with mitochondrial neurodegeneration. So here you have more demand or less supply, your mitochondria do not work, you cannot lift the ATP need that is required. In DNA repair deficiencies, the mitochondria seem to be okay actually. And membrane potential is even increased, but the demand seems to be much higher. So even though you're trying to compensate, you cannot meet the demand of the brain. And so to recap, you have DNA damage in the nucleus, you get PARP activation, and you cannot shut down PARP, which means that you're losing ATP and NAD, and then you get mitochondrial dysfunction. So what about people? So actually we know that with age, weight loss is probably not good for you. So when you reach 70, it's good. So your lowest mortality risk is actually at about a BMI of 27, which is a BMI that would be associated with overweight when you're younger. So this could suggest that you need to increase your body mass over time. And in fact, if you look at your risk of developing BMI, your risk of developing dementia in the next 7 years as a function of BMI, then I think quite strikingly, the higher your BMI is, the lower your risk of developing dementia is actually. We just have to remember here that these are people that that do not have diabetes. So if you get diabetes, then your risk of developing dementia is very high. But just being overweight by itself appears to offer some protection for your brain. And in fact, just increasing fat intake actually decreases your risk of death. If you look at total mortality, and if you get 45% of your energy intake from fat, your risk of dying is 30% lower than if you get 10% of your energy intake from fat. And vice versa, if your energy intake comes primarily from carbohydrates, then your risk of dying is increased. So we all know this dilemma. You're in the bakery in the morning, and you have to choose if you should take a croissant or a Danish. And it's always a difficult choice. But in fact, you should probably choose just a cup of coffee and maybe some dark chocolate with very low sugar composition, which includes quite a high amount of fat and probably healthy polyphenols. All right, so that was one take home: eat more dark chocolate and drink more coffee and eat less sugar. The other take home message is, of course, that DNA repair is important in aging. And so we were considering one way to possibly be able to stimulate DNA repair. And the way we thought about this was to take advantage of something that Nietzsche actually came up with more than 100 years ago, which is, what does not kill me makes me stronger. And in cells, we know this phenomenon is called the hormetic response. So if you treat cells with a certain type of stressor, then the cells become more resistant to the same type of stressor and similar stressors on subsequent exposures. So there's an adaptive response from the cells that allows it to better deal with this type of damage. So we thought about, can we figure out a way to make the cells think that it has received a DNA damaging insult, so it will upregulate its DNA repair pathways in the future. And the way we did that was through a collaboration with Silicon Medicine, where we used deep neural network to find compounds that had a hormetic signature. And so we screened actually more than 15,000 compounds and got a number of hits here. The first question was to understand if DNA, if these drugs induce DNA damage, because we did not want the drugs themselves to induce DNA damage. We just wanted them to induce a response that looked like a DNA damage response. And for this, we collaborated with Amelia Technologies and used their Comet chip technology, where you can screen for DNA damage in cells. So here's some examples of data for this Comet assay. It's basically you embed the nuclei of a cell into agarose, and then you have the nuclei embedded in this gel, and then you run an electrophoretic field across it, and the DNA will then be pulled out of the nucleus. This is why it's called a comet assay, because these ones look sort of similar to comets. The longer the tail, the more breaks you have in the DNA. So clearly some of these drugs do elicit DNA damage breaks, but many of them actually do not. So another way to look at this was to look at if they induced DNA damage. And again, just to remind you, there are DNA damage markers such as gamma H2AX and 53BP1, which indicate double-stranded breaks. And looking at these markers in the number of compounds that we tested, we saw that some of them did induce a DNA damage response by gamma H2AX and 53BP1. Many of them did not. And so we then turned to a more functional assay. If the cells or if the drugs should induce a hormetic response, the cells should become more resistant to ionizing radiation. And indeed, this is what we see. We see some cells are actually completely resistant to a very high dose of ionizing radiation. And so we tested some of the drugs to see if they would affect a cellular aging assay. This is just an assay where you look at cellular replication. So how many times can a cell divide until it goes into the senescent state? And you can see that this drug F seemed to be able to allow the cells to grow more. And this is this is a very, very long experiment. Many weeks in culture. And so this was quite extensive. Excuse me. So we also wanted to see if the cells developed senescence. And for this, we used beta-gal staining and a deep learned predictor that can find beta-gal and predict senescence in cells. And using this approach, we can see that that this drug decreases the amount of senescent cells, suggesting that it reduces senescence. And not only does it reduce senescence, it actually appears to reverse senescence. So if you look at one of the features of senescence is the cells stop dividing, which means that they stop expressing a protein called pCNA, which is involved in DNA replication. But if we treat with this drug F, we can see that there's a rescue of pCNA levels almost almost similar to what's in wild type. And you get actually cells also start to grow. So this was quite remarkable. Drug F also reduces the amount of DNA damage in replicative senescent cells. So there's less DNA damage foci. These are the persistent foci that I mentioned in the beginning of my talk. And you actually see a reduction with drug F. And this was in the repetitive foci, repetitive senescent cells, but also in the IR-induced senescent cells. So if we treat cells with ionizing radiation, wait 7 days, the cells become senescent and are then filled with these DNA damaged foci that will not go away even if you wait up to 3 months. However, if we treat with drug F, you actually reduce the amount of foci to what's comparable to normal levels. So to test if these drugs work in vivo, we turned to an animal model that's used in aging, and this is the fruit fly. So we have a spin-out company, Scheibye, where we can do high throughput testing of drugs in animal models. And here we tested in fruit flies. So there's an AI that can track these flies over time, and we can then collect behavioral data on the flies and automatically detect their lifespan and their health span. And doing this, so here, each line here is a separate fly path. When you're young, you walk quite a lot. When you're young in drug F, you also walk a lot. If you're old, you walk less. But if you are old on drug F, you still walk a lot. This is quantified here. So this decrease in gait over time is actually quite conserved over different species. So in humans, for example, a decrease in gait speed is one of the strongest predictor of mortality in the next 5 years. And this is the same also in mice. So, and this is probably because walking is actually quite complex phenomenon. You need your brain to interact with your feet, and you need balance and so forth. So it gives a good view, overall view of how the system is working. So if you start walking slower, then your system is not working as well as it used to be. And so this is also the case in fruit flies and Drosophila, but if you treat them with drug F, you preserve this. And you actually increase the lifespan of the flies in drug F. And so right now I just want to highlight this, that we are looking for analogs of drug F to find more potent compounds. And we have tested a number of found a few. And with that, I'm gonna stop my talk. I hope that you enjoyed this introduction to DNA damage and aging and how we can potentially interfere with this response to preserve our particular brain function with age. Thank you very much for listening.